Hi everyone, welcome to my channel Pure Biology, and today I come with another video, that is the nucleotide excision repair. This is the second part of the excision repair. The first part, that is containing the base excision repair. So I have already explained in my previous video, so you just check out. And this is the second part of the excision repair. This is the nucleotide excision repair. The main difference of the nucleotide excision repair and the base excision repair is that here the portion that containing the damaged bases, that is totally cut off from the strand, and new strand containing the correct bases, that is attached with the or pairing with the another strand. Okay. So here the main important thing is that how this is happening. The repair system includes the breaking of the phosphodiester bond on either side of the lesion. Okay, the phosphodiester bond is cut off from the two part or two sides on the same strand, not the both strand, only the strand that containing the damaged bases. This is cut off from the both sides. Okay. And this excision leaves a gap which is filled by the two enzymes functions, that is the ligase and the DNA polymerase. Okay. So here this is an example, is that UVR system. This UVR system, this is a great example of the excision repair, and it is found in the E. coli. Okay. So here actually the strand containing 12 nucleotides in length, this is removed. 12 nucleotides containing, that is the capability of removing the strand which is containing the maximum 12 nucleotides. Clear? So here the main important genes, that are UVR A, UVR B, UVR C, these are main important genes which has which have the products. These products actually help in this excision repairing process, and called this product or this enzyme is the ABC nuclease. The enzyme actually binds to DNA at the site of the lesion. And first UVR A and UVR B attach to the DNA at the damaged site. So which must be bind at the damaged site? That is the UVR A and UVR B. Okay. And this UVR A's function is that it can recognize the damaged site. What is the function of UVR A? It can recognize the damaged site of the DNA strand. And the departure of UVR A, it allows UVR C to bind, forming UVR BC. So first of all, when after the recognition of the damaged site of the strand, the UVR A that is released from the strand. So which can bind at that place? The UVR C, and this bind with the UVR B to form the dimer UVR BC. Clear? And the damaged strand generally at the site 8th phosphodiester bond on the 5' site of the lesion, and at the 4th or 5th phosphodiester bond on the 3' site. That means suppose this is the 5' site, this is the 3' site. So the cut must be towards the 5' site. This is the 8th position, and the cut must be towards the 3' position. This is the 4th or 5th site. Clear? Now UVR D. What is the function of this UVR D? This is the helicase, and this helicase actually, what is the function of the helicase? You all know that it can help to unwind the DNA double helix. So UVR D, this is the helicase that helps to unwind the DNA to allow release of the single strand between the two cuts. So its function is to help the cut off the or removing of the strand that is already cut by the enzymes. Okay. And now resulting gap is filled by the DNA polymerase and sealed by the ligase. So DNA polymerase function is, it generally you know, the extension of the DNA strand. This is done by the DNA polymerase. So its function is that it tries to make a bond between the bases, and ligase function is that the seal the base pairs with the strands. So in the two enzymes, DNA polymerase and DNA ligase, help in the completion of the strand, or they try to fulfill the strand completely. And in human, the one important example, this is the xeroderma pigmentosum. This is caused by this type of excision repair. And this is the rough diagram where you can see first TT dimer is formed at the 5' to 3' strand, and here UV or AB binds at that position, and now A is released from this position and C tries to bind with the B and form the UV or BC compound, and after that the total part it is removed from the strand, and in the next part the two enzyme polymerase and ligase, they complete the strand with the proper base pairs. Okay, and here in the cutting of the strands, that is possible by the enzyme is polynucleotide. Okay, UVR BC, not polynucleotide, UVR BC cuts the polynucleotide. So main role, the cutting of the strand, it is found the main role belongs to the BC compound. Clear? So this is the excision, nucleotide excision repair. I will try to explain another repair system, that is the mismatch repair system, in my next video.